And Friday, tomorrow, 2 o'clock, Olin Theater, research presentations, opportunity for you to earn extra credit. If you come and then submit a short write-up about how presentations could be related to this. I think there are some physics presentations and some presentations that may not be as directly related. That's fine. But use your creativity. All right. I am happy to answer questions people may have, but I was also thinking that these are the things, the major concepts that we covered, right, in biological psych. And I was thinking I could just sort of run through the major concepts of those, and if you have questions along the way in there, you can sprinkle them in. Does that sound okay? Yes. And if we have time at the end for additional sorts of questions, and plus we have a review session that's optional next Monday, 12:30, if you're interested in that. If you meant to print out a list of who has already told me they want to take the test early to check on that, but I forgot. So, if you want to take the test early, that's fine. Just maybe double check with me that you have said that, so that I'm sure that I'll be here and ready to give it to you when you want to take it early. And any of the exam periods before you're here. Okay? How many people want to take it early? Just show hands. 1, 2, 2. Okay. No problem. Have y'all already contacted me? Some people have.
She has, I have not yet. Okay,
fine. Then I have Sam there and I'll get you. No worries about that. All right. Where do we start off in here? Neural signaling. Way back when. And how important is neural signaling? All this other stuff relies on that, right? I mean, it's the foundation. It is the foundation, right? So, and I'm going to post these slides afterwards, right? And I can take a picture of this and post that in my next notes up there on that. All right, so neural signaling. I drew this purple dashed line in there. Why did I draw this purple dashed line in there? What's that represent?
When it switches to, right? Something changes.
Something changes.
Like the release of the neurotransmitter.
Yeah. So fundamental difference on this side of the purple line, all of the potentials are synaptic potentials, and they are created by neurotransmitter-gated channels. So neurotransmitter has to bind to open the channel, right? And there's 3 different major types that we talked about. Sodium, which creates EPSPs. Potassium, which creates IPSPs because the positive potassium leaves, right? Chloride, negative charge enters, it's an IPSP. Right? And this is the property of the dendrites and soma. Synaptic potentials, neurotransmitter gated. And if the EPSPs summate through either temporal or spatial summation and reach threshold, -50, then we have a fundamental, at the axon hillock, a fundamentally new process involving voltage-gated channels. Now, the -50 voltage is what opens the channels. And we have 3 channels that are involved in that. 2 are involved in the action potential. That's the transmission of the signal down the axon terminal. Sodium opens at -50, allows the rising phase, sodium comes in. So much sodium comes in, it goes past 0, all the way up to +30, and it closes. Rising phase of the action potential. As that action potential goes past -40, potassium channels open. Potassium starts leaving. And it starts leaving even faster once it gets more positive inside of the neuron, because now both electrical and chemical gradients want it to leave. Right? As potassium's leaving, because it's positive, it's pulling that charge back down. creating the falling phase. So much potassium leaves, goes all the way back down to negative 75, below the resting potential. And now, all the potassium is where? Where did all the potassium go? Outside. Where did all the sodium go? Inside. Is that what we want? Do we have any forces built up to start another action potential? No. So we have a refractory period where the sodium-potassium pump has to work to restore all those forces. That uses energy, right? It uses energy, and it's a time that we can't have action potentials. But as it rebuilds those forces and comes back to resting potential, now we can regenerate a new action potential. What's probably not shown as well here is we have myelin insulating the axon, and so this action potential is happening at each node, and the sodium is spreading out down to the next node to trigger the next negative 50 voltage gated channels, and it spreads and it spreads and it spreads. When it gets to the axon terminal, we have our third type of channel, right? The calcium channel that's voltage gated still. So the voltage of the action potential opens the calcium channel. Calcium comes in, activates synaptotagmin, causes neurotransmitter release. And then there's three things that happen after that. We got to get rid of the neurotransmitter through either reuptake, which is recycling, bring it back in, reuse it. Break it down with enzymes like monoamine oxidase or acetylcholinesterase, or the glial cells can absorb the neurotransmitter and get rid of it. Cool? All there. Along the way, we talked about disorders that are associated with that. What disorder are we talking about with neural signaling? Well, first of all, with neural signaling, what are the four major parts? Input. What's the soma do?
Processing.
Right. That's summation.
And then transmission.
Uh-huh.
And output.
Right? We just went through all those. So what disorders associated with it? MS, right? Autoimmune disease attacks the myelin, you don't have myelin, and that stops the flow of those action potentials.
What is it attacking?
Hmm?
What is it attacking again?
The autoimmune disorder attacks myelin, so it removes the insulation, and the sodium can't get down to the next node. It flows out of the axon before it can trigger the next regeneration, saltatory conduction. All right, development was the next thing we talked about. What do we talk about with development? Talked about sort of 6 stages of development. What were they?
Proliferation.
What's that?
One was proliferation.
Yeah. It's actually over proliferation.
Isn't apoptosis the last one?
Yes. Migration, then what? Differentiation. Then what? Apoptosis. And then we had two more, which was what? Yes, myelination and synaptogenesis. Right, something like that, right? Forming synapses. Overproliferation, right? Make more neurons than we're going to need. Those neurons migrate on radial glial cells. Then they release from the radial glial cells and use neurotrophins. So neurotrophins was a key concept. The neurotrophins both guide the neuron to its final place and cause it to differentiate. It causes it, the neurotrophin causes it to differentiate, Grace.
What does that last one say?
Synaptogenesis, creation of synapses. And then once it's in place, differentiated and connected, if it's in the right place, the neurotrophins then support its survival. If it's not in the right place or not connected in the right way, then it undergoes programmed cell death, apoptosis. Calcium comes in, activates diabloprotein, and it dies. Again, calcium, important signaling molecule in neurons. We've already talked about it causing neurotransmitter release. It can cause apoptosis. It can cause things like an LTP for more receptors to be added. So calcium in general is an important signal for neurons. Same.
Weren't there like 3 different ways they can migrate, or was this where we talked about like 3 different types of glial cells? I remember talking about 3 of something possibly.
No, there were 2 ways that it can migrate. On radial glial cells, out to where it's supposed to be, and then it detaches and follows the neurotrophin signal. All right. Developmental disorders we talked about. Yeah. ADHD and autism. All right, next we talked about the motor system. In the motor system, what was an important part of the brain? The basal ganglia, yes, the basal ganglia, right? And so we talked about the normal circuitry. We all learned how to draw the boxes, 4 boxes, a box below, a thalamus output, and a substantia nigra input, right? Excitatory coming in, excitatory coming out. Everything else in here is inhibitory except the subthalamus coming back in, right? 2 dopamine inputs to the caudate putamen, excitatory to the putamen and inhibitory to the caudate. What's the basal ganglia responsible for? Regulation of motor control and what? Regulation of cognitive thought, right? And so what disorders did we talk about for motor system?
Parkinson's.
What else?
Obsessive compulsive disorder.
Yes, because of thought, OCD, and we also talked about NPH, normal pressure hydrocephalus. Right? So in Parkinson's disorder, what happens? This goes away, right? We lose dopamine input. So then you just follow the path. What happens if you take away the inhibition in the caudate?
It increases.
more output, and it's inhibitory. That means it shuts down this and decreases its inhibition. If we decrease the inhibition to the subthalamus, what happens to it?
More output.
It gets excited, right? And so we're now exciting the internal globus pallidus. So big inhibition on the thalamus. We take away the excitation of the putamen, and what happens?
More.
Less signal, right? And so now we have a big output of inhibition, and we get decreased responses back to theirs, which results in what kinds of symptoms for Parkinson's disease? Inability to control movement, particularly inability to initiate movements, so shuffling, lack of arm movement when they're walking, inability to suppress tremors, so they get tremors that they can't suppress, right? And eventually, it hurts their cognition and becomes hard for them to think, hard for them to generate thoughts.
Doesn't one of them get mistaken for dementia or Alzheimer's?
Normal pressure exercise. Huntington's disease, what happens?
The cognate is compromised or dies or something.
Yeah, so for disorders in general, what are the symptoms? How's it diagnosed? What is the cause? Right? If known, right? What is the biological mechanism or correlate? What are the treatment options? So that's what you should know for every disorder, right? What's the cause of autism?
We're not sure.
We're not sure. We think there's evidence of genetic influences. It affects boys more than girls. We see if one male sibling has it, there's a higher prevalence that another male sibling may have it, so there's some familial lines. It's not purely genetic, but there's a genetic influence. But other than that, we don't know. It looks like it happens very early on, but we don't know, right? So sometimes we're not going to know the cause, right? Here, sometimes we may not know the biological mechanism, right? We may just know what's correlating in the brain. You can look at the brain and see this, right? So for Alzheimer's disease, we can see amyloid beta 42 plaques and neurofibrillary tangles. What is the mechanism that causes that? We don't know. But that's what's going on in the brain, loss of neurons in the hippocampus, right? We can see that, okay? All right, so what's the cause for Huntington's disease? That's the mechanism. Yeah, we know for that one, there's a dominant mutation on chromosome 4 that causes production of Huntington protein that destroys the caudate. Destroying the caudate is the biological mechanism, right, that's causing this. Right? Treatment options.
Is this the one where there's like surgical implants in the men's shoulders and like electrical regulation of the brain?
Yeah, that was for
Sorry.
for for Parkinson's and for, we also mentioned Tourette's here. Sometimes we don't have a treatment, right? There are some drugs that can help reduce the chorea movements, the wild movements that are associated with this, but there's no treatment to stop it from happening. People die from it, right? So sometimes we don't know the cause, and sometimes we don't have a treatment, unfortunately. Along with this, right, we talked about the role of dopamine. Right? We can add in things that we learn later, like schizophrenia, psychosis, right, here happening as well. We want to be in this normal range. When we're too low, just below, we might be in ADHD land. All right. Obsessive compulsive disorder involves a circuit between the orbital frontal cortex, which is the obsession, right, and the basal ganglion, which is the compulsion. Right? It's the motor cortex, the compulsive behavior, that can eventually stop the cycle. Right? But that's not what we want, because we don't want the person to be treating their obsessive thoughts by washing their hands constantly all day long. Right? So we have to have some other interventions. And so there are 3 different types of treatments, right? We can block dopamine here to try and reduce the activity, to stop the circuit. Kind of effective. We can do CBT to unlearn the association between the compulsion and the obsession. Very successful if the patient buys into it. If neither of those are successful, we can surgically sever the connection and break the connection physically. Yes?
We may not need to know this for the exam, but when we were doing split brain patients, you talked about the consequences, even though it's used to treat epilepsy. What are the consequences of severing this connection with surgery?
I don't know that we know that. I don't know that we know that. It's a much more precise surgical manipulation than severing the 2 halves of the brain, right? All right, sensory systems. The things we need to know about the sensory systems, right? We need to know stimulus, transduction, neural code, which is label line or cross fiber pattern, pathway, and CNS areas. All right? Broke it down for you here. Questions about that? That's just information. Yes?
Can you briefly go over the labeled line and then cross fiber pattern, and then specifically with vision?
Sure. So vision is the most complex of all the senses. So I just gave you 2 examples of label line and cross fiber pattern. The thing you need to understand about label line and cross fiber pattern, label line means that a neuron, when it's active, represents a single stimulus. So the brain only has to know, is that neuron on or off, to tell if that stimulus is present or not present. Okay? So the example for label lines was trichromatic and opponent process theories, saying trichromatic, if long wavelength cones are active and those neurons are active, that means something is stimulating the red in the long wavelength part, right? Could be red light, could be some other thing that we have to look across the fibers to understand, right? So we have long wavelength cones are labeled line, medium wavelength cones are labeled line, short wavelength cones are labeled line. They form excitatory inhibitory connections to create a opponent process. Blue on, yellow off, right? Now we're getting into having to look at across fiber patterns. That really belongs with that. Because now we have to look at the pattern of action potential. So if we have to understand the pattern across labeled lines or the pattern within a labeled line, that is a cross fiber pattern. And these things work together. It's not one or the other often. They work together, right? So if I want to know if it's yellow light at the trichromatic receptor, short, medium, long wavelength cones, what would yellow light stimulate? There's not a yellow cone, so what would yellow stimulate?
Green.
Equal amounts of medium and long wavelength. It's in between medium and long wavelength that would activate them equally, right? So the brain would have to look across the label lines to say, hey, both, it's not just long, that would be red, it's not just medium, that would be green, they're equally activated, that's yellow, right? So that's an example of a cross fiber pattern versus label line, okay? So anytime there's two signals, we have a baseline firing rate and the neuron can either be excited and it means one thing or be inhibited and it means another thing, that's a pattern. That's an across fiber pattern. Okay, that's pattern encoding. And the brain has to count action potentials to know what the signal is. It can't just know it's on or off. Cool? Other sensory questions? All that's up there for you. Yes?
For the auditory one, what range is human voice again? You said we're better at hearing that than anything else.
It's 500 to 5,000.
Okay.
We have the biggest number of receptors to it.
Okay.
Right? All right, that brings us to homeostasis systems. We talked about body temperature, thirst, feeding, and sleep-wake cycle. Right? Those are the four big hypothalamic systems that we talked about. They have specific nuclei associated with them that do specific functions. Feeding being the most complex. Good. So what did you need to know for homeostatic systems? What were the things? We need to know set point, receptor, the nucleus, and the output. Cool? All right. Feeding. We talked about short-term signals coming into the hypothalamus and long-term signals coming into the hypothalamus. Long-term signals being our fat reservoir. When we have a lot of fat, we have a high amount of leptin. When we have a low amount of fat, we have a low amount of leptin. Those two things stimulate two different areas of the hypothalamus, right? That are going to affect how big our meal sizes are. If I have low fat cells, I want to eat bigger meals. So I'm going to inhibit the PBN, and when we turn the PBN off, meal size goes up. Right? If I have a lot of leptin, ideally I want to excite the PBN. Activity in the PBN turns off meals, shuts down meal size. Short-term signals. Ghrelin builds up. The longer we go without eating, the more ghrelin we have. Ghrelin acts, again, through the arcuate nucleus to inhibit the ventromedial nucleus, which is going to increase the number of meals. So if I haven't eaten, I want to start a meal, right? High ghrelin, start a new meal. Right? I'm going to increase numbers of meals. If I have a high amount of the satiety hormone, PYY, it's going to do the opposite. We're going to shut down meal size because I'm full. Yes?
Which of those chemicals did pharmaceuticals think was going to be the key to weight loss, but didn't end up working?
Leptin, right? They thought, hey, we could just give, we could just act on the leptin system, right, to shut down meal size, and people will lose weight. It doesn't work that way. Yes?
Will we be able to draw pictures like we have in the past?
Yeah. You do not have to write complete sentences. You can do bulleted points. You can draw a diagram, label it, say this is this, this is this. Just convey to me that you understand the material. Really, truly, this is your opportunity to show me all that you've learned. I know you've learned a lot in here. All of you have, right? And so this is your opportunity just to demonstrate that. Think about it that way, okay? It's a final opportunity. All right.
About the word final.
Well, it's only final because I'm not going to come back in January and ask you to do it again. All right. Sleep-wake cycle. We kind of broke into two parts, right? So we talked about circadian rhythms when we talked about the hypothalamus, because the suprachiasmatic nucleus, the clock, and these genes, right, are involved with helping us regulate our 25-hour sleep-wake cycle with light coming in and resetting that each day to 24 hours, right? But then when we talked about learning and memory, we talked about sleep cycles and the brain's control of our sleep-wake cycle, of going to sleep and waking up. And so that's kind of laid in over here. So we have the pons with the raphe and serotonin for sleep acting up here. We have adenosine as sleep pressure in the basal ganglia acting. So we can kind of put those two things together. This sleep-wake stuff. I talked about it there because of the importance of slow-wave sleep and REM sleep in learning, in processes of learning, different processes of memory consolidation, right? And I wanted to put it with that after you learned about memory consolidation, right? But we can talk about this all together, and GABA then putting the brain to sleep, and over here we have norepinephrine and acetylcholine and histamine coming in to stimulate being awake. And then we have this whole cycle here contributing to that along with the pineal gland and melatonin. Tracy?
Would you want us to be able to talk about those different, like the REM sleep and all of that?
I do.
on the exam?
I do.
Okay.
Particularly, so I'm kind of jumping down to here, right? Particularly with regard to and really it's that if you would focus on slide 21. That sort of summarizes the differences between slow wave sleep and REM sleep. All right, we're good on that? Cool. All right, yeah, we're gonna skip over that. So, stress, HPA, immune systems, right? Really need a slide on that. I will. I missed that. I'll add a slide into this that will have this whiteboard image on it. I think pretty much this slide, right? I'll add a slide in there in between the circadian rhythms and when we talk about learning and memory that covers this. But right, so we have HPA axis. Hypothalamus, PDN, anterior pituitary, adrenal cortex. CRF causes ACTH, causes cortisol throughout our entire body from the adrenal gland. That's our stress response. Normally, an adaptive stress response, we have a physical stressor. We gear our body up to deal with it, we deal with it, or we die, and then it's over in 20 minutes. 20 minutes of terror, and then you're either good or gone. All right. And then it's designed to shut itself down in 20 minutes. The cortisol buildup turns off this, stops releasing cortisol. It's an automatic turn-off system. It takes about 20 minutes. The problem is psychological stressors override that. And they keep coming in and saying, no, you can't turn off. Keep producing cortisol. Ignore that negative feedback signal. Right? It's when the normal adaptive response to the physical stressor, we escape, right? So then that's no longer causing this, then it can be turned off. But if we have something continuing, or if the physical stressor is this horrible roommate environment that you have that you can't escape, that's constantly there, right? You can have physical stressors that don't go away, right? But this was designed for more primitive species, fight or flight. I come in contact with something that's going to kill me or injure me, I got to get away, or kill it, or die. 3 options, right? And the stress system was sort of designed to help that. It becomes maladaptive when we use it chronically. It needs a lot of energy, and so it hurts the immune system. Because it steals energy from the immune system when it's chronically active, causes our white blood cells to go down, right? Causes us to become more susceptible to becoming sick when we come in contact with bacteria and viruses. If we have, the other way it can affect the immune system is if we are constantly turning it on and then turning it off. So we stress and then we let it go, and then we get stressed again, and then we let it go, and then we get stressed again. It starts to build up and it doesn't return to baseline, and the immune system can become hyperactive and we can get autoimmune disorders. Right? So here we talked about, particularly with stress, we talked about PTSD, right? And how some individuals that have more of these receptors would be more susceptible to a big stressor happening on the system than individuals with less, right? And we talked about autoimmune. That's chronic intermittent stress. So chronic stress causes immunosuppression. Chronic intermittent stress causes autoimmune disorder.
And for the fifth, for homeostasis systems, would a disorder be the proper immune system?
Yeah, or obesity, or diabetes, different things like that. All right. Next up was attention, and I don't really have a slide for attention, but I'll also do emotion. So for emotion, the emotion system, we talk about the limbic system, right? And we talked about the role of serotonin in the limbic system and the role of the amygdala in the limbic system. And so here we talked about the amygdala, the limbic system, and from a disorder perspective, we talked about major depressive disorder, postpartum, bipolar, seasonal affective disorder, and anxiety. Main things that we talked about there, right? Attention, what do we talk about with attention?
Wait, what's SAD?
Seasonal affective disorder. Depression associated with winter. Where in the brain are the attention centers? Frontal lobe for working memory or short-term memory, and where else? It's lateralized. What does lateralized mean? Yes. So which parietal lobe?
Left.
Right. What's in the left parietal lobe? Language. Right? Right parietal lobe, right? Involved with our conscious awareness of our world. Right? Our conscious awareness of our world. What's the disorder associated with that? Contralateral neglect, when you damage that right parietal lobe. And that is a left world disorder.
So parietal lobe is language and the right is attention.
Yes.
So you can language.
Let me, can I hang that block paper? I'm gonna shift gears this other way. Language. Let's talk about what structures are involved with language. All right, that's our brain. Oh, this is all messed up.
Focus area.
All right. So, what are the areas involved with language? Wernicke's and Broca's area. That's a good place to start. Where is Wernicke's area? Left parietal lobe, right? And where's Broca's area? This is a central sulcus. What is this? Post-central, and what is it? What does it do? And what does the one in the pre-central do? Right? And then we have a supplementary motor and a premotor cortex in front of that help coordinate stuff. Where's Broca's area? In the premotor area. How do we get from Wernicke's area to Broca's area? We got to go under this. We got to go under this, right? Arcuate has to travel under that to connect them. Right? What's our input into language? The auditory, so where's the primary auditory cortex? Superior temporal gyrus. So we have, that's an ear, in case you don't know. Right? And it comes in both sides, right? There's another one over here. Comes in and has to cross over. I don't even know how to draw that. It's one-dimensional.
Which lobe is the Broca's area?
It's kind of the junction of parietal, occipital, and temporal.
No, Broca's.
Oh, Broca's, front one, right? Everything in front of the central. Sorry. Right? So that takes the left ear comes in, the right ear crosses over, goes to the left auditory cortex. Right ear comes in, left ear comes in, goes to the right auditory cortex, has to go through the corpus callosum to get over here to Wernicke's area, right? So we have input from both ears. When it's language, gets consolidated in Broca's area on the left. I keep saying Broca's area, I'm sorry, you guys correct me. Wernicke's area. Comes into Wernicke's area, right? It's a lot. What does Wernicke's area do?
Language comprehension.
Language comprehension, right? If there's a stroke that affects Wernicke's area, what happens?
Aphasia.
We get aphasia, and what are the symptoms of Wernicke's aphasia?
You can speak fluidly, but like you can't remember names and words and stuff like that.
Yeah, so you're able to speak and communicate, but you have a hard time understanding other people, understanding commands, and you have a hard time knowing when you're making mistakes because you don't have the feedback to know that you're saying inappropriate words.
But they understand what's being said to them, they just can't comprehend their own thoughts.
No, sometimes, it depends on how severe it is. Sometimes they have a hard time comprehending what you're saying, right? They can hear the words, but they can't, put them together. All right, from there it goes to Broca's area, right? Is there another input into Wernicke's area? Yes, right? Right? So when the visual system recognizes, so we have an oral and a reading, right? A heard and a reading input. Both of those end up in the same place in Wernicke's area, right? What about outputs? What does Broca's area do? Language production. And how many language production outputs do we have? What are our language production outputs? Speech, right? Which is going to go here. Speech output. How else do we have language output? Writing, right? Writing is going to have to go to but this is the left hand, so most of the time it's going to have to go to the right hand. No, I did that wrong. Dang it. That's what I did. To the right hand on the left side of the brain, and people that are left-handed has to go over to the left hand, right? If we're writing with the left hand. That's right. Good? On language. I'll take a picture and post that. Don't worry about it. All right. Lastly, learning and memory. Right? We talked about different types of memory. Iconic or sensory memory. That's happening in our sensory perception areas throughout the brain, wherever those are. Right? We talked about short-term or working memory, dorsolateral prefrontal cortex. Dorsolateral prefrontal cortex, short-term working memory. From there, we get into our different types of long-term memory. In general, for explicit memory, we have the hippocampus is encoding those memories and they're being stored in the temporal lobe. Semantic is information, declarative is episodic, what happened. We also have procedural memory, implicit memory. Procedural memory, the cerebellum is involved with that, not the hippocampus. HM could do procedural memory. He could form procedural memories. He couldn't remember that he had formed them or had done it, but they were there because his cerebellum was intact. Spatial maps. Those are actually stored in the hippocampus. Then we have associative, classical conditioning, operant conditioning. Those are going to happen. The cerebellum, we know, plays a role in that. So these are non-hippocampal things. Cerebellum plays a role, and then whatever areas of the brain are specifically involved. If I'm ringing a bell and presenting food, then the auditory system and the food stimulating systems are the ones that are forming connections with each other. If I'm blowing a puff of air in your eye and ringing a bell, then the auditory and the eye reflex circuit are what's forming strong connections. So it depends on what the learning is as to where it happens in the brain, but it's non-hippocampal. It's non-hippocampal. Okay? And classical conditioning and operant conditioning, classical conditioning, two things constantly together become associated with one another so that one can trigger the other's output. That is a form of postsynaptic learning, strengthening connections, like with LTP, but not requiring the hippocampus. Operant conditioning, behaviors that are reinforced, increase in frequency, behaviors that are punished, decrease in frequency. And then we talk about non-associative learning, habituation and sensitization as presynaptic changes where we either decrease the release of neurotransmitter or increase the release of neurotransmitter in the presynaptic side. Again, depends on what circuits we're acting on as to where that is in the brain. There's not a specific area that controls that. Cool? Learning and memory? You guys are like, this isn't that bad. We can cover everything in the semester in 80 minutes. LTP. Right? This should be fresh, literally fresh in your brain. Right? Presynaptic side, postsynaptic side, changes involving AMPA and NMDA. Again, calcium, important signaling molecule to trigger more AMPA receptors being put in that synapse, which makes it stronger, so it doesn't have to work as hard. So we get a retrograde endocannabinoid signal, and we can now release less neurotransmitter to get the same response as we had before. Important point of this is repeated use. You can't get LTP without bumping off the magnesium, and that requires repeated stimulation. Good? All right. Disorders. Again, these are the four major things: symptoms, cause, mechanism, or correlates, treatment options. We talked about autism and ADHD, basal ganglia, those four. Stress, PTSD, immune disorders. Emotion, anxiety and mood disorders, major depressive, postpartum depression, bipolar, seasonal affective disorder. Attention, talked about contralateral neglect, left side world neglect. We talked about aphasias, Wernicke, broken, and conductive aphasia. When we sever, we have a damage with the arcuate fasciculus with conductive. We have to relearn, we have to have rehabilitation to reconnect those functioning processing areas. And then we talked about a bunch of sort of cognitive disorders like stroke, the role of the penumbra, right? Dementia, the ability to differentiate between Pick's disease and Alzheimer's disease and normal pressure hydrocephalus. Schizophrenia, we just talked about. Right? What's the cause of schizophrenia? It has genetic influences. It's not 100% genetic, but in monozygotic twins, there's a 50% correlation. Right? And we know that it's something that happens probably in uterine, in prenatal, early in development, that is a long-lasting effect. Viral exposure is correlated with it, right? Fevers. But we don't know. Like, we know some. And if I ask you about a disorder and the cause, I don't want you to go, we generally don't know. Genes and we don't know. I want you to be more specific than that, okay? That's not going to be a good answer, okay? What's the evidence for genes that we know, right? Monozygotic twins show genetic evidence, right? Viral exposure to viruses and fever shows environmental, in uterine environmental effects. But we don't know the total picture. That's okay.
For postpartum depression, can you go over the causes of that?
Yes. So loss of pregnancy, and so immediately after giving birth, there's a sense of loss of the pregnancy and changes in hormonal levels, trying to get back from a pregnancy hormonal state to a normal cycling hormonal state, and stress. Right? So there's physiological and environmental effects on that.
Is hormone cycle need to be on our list, our master list?
No, that wasn't sort of a major thing that we covered. The reproductive stuff was kind of... This is what I want you to focus on. I would use this as review points for the final. Okay? We don't exactly know the biological mechanism for schizophrenia, but we can look in the brains and see what?
Where there's activity, excess dopamine.
We can see lower activity, which we think is involved with dopamine because blocking dopamine improves the symptoms. So a lot of times we say, hey, dopamine is a correlate or a mechanism because treating it helps, right? Serotonin is a correlate because treating it helps, right? But it's not a chemical imbalance, unlike Parkinson's disease, right? And we know that there's disorganization of cells in the hippocampus and a loss of cells in the brain. All those things are things that we can see in a schizophrenic brain. Treatment options for schizophrenia. Medication. What about CBT? It can't treat the disorder, but it may help the patient understand the disorder. It may help with patient compliance with taking the medications. It may help with other issues, right? But it can't treat the disorder. Good. All right, and then lastly, addiction. What did we talk about with addiction?
Alcoholism is most popular.
I didn't hear.
Like alcohol and
We applied it. What did we talk about in terms of theoretical models?
Disease model.
Yeah? Disease model involving influence of genes.
Dependence.
Physical dependence, we're talking about tolerance and withdrawal.
Psychological benefits, positive.
Positive reinforcement or environmental effects. Escaping stress. Escaping negative living conditions. Right? Typically. The euphoric high. Self-medicating for depression. Right? Then we applied, we understand like the basis of these, and then we can apply them when we did that as an example with alcohol. Right? Is there an effect of genes on alcohol? Do we see familial alcoholism? Yes, particularly in Males, right? What about physical dependence? Do you get tolerance and withdrawal with alcohol?
Yes.
Yes, right? What about positive reinforcement? Why do people use alcohol?
Yeah, take stress off the field.
Right? For the positive benefits of it, right? Yes?
For tolerance and withdrawal, is it for alcohol, is it more of a tolerance than what are withdrawal symptoms?
Tolerance means that the body adjusts so that you now need more alcohol to achieve the same effect,
Right.
which that happens for sure.
For this part.
And withdrawal means when you take away the alcohol, there's a negative rebound. And that certainly happens as well.
Like a hangover?
No, it's much worse than that for alcoholics. They can have seizures, they can have cognitive deficits, they can feel sick for a week or 10 days. That's why they often have to go to a treatment facility to get care.
We talked about how some people will willingly put themselves through withdrawal, so that way they never want to touch the stuff again, but that doesn't always work. Why?
Because they forget.
They forget.
And the reason that they use it becomes stronger than the memory of how bad it was. Right? Because they may go through that, but they don't go to therapy, so they don't resolve whatever kind of issue was causing them to drink that much in the first place, and then they resort back to it. All right, and then we talked about each of those three in terms of prevention and treatment. What does the disease state say about treatment and prevention?
Abstinence.
Abstinence. You have an addictive physiology, right? And so how do we deal with that? I don't have a magic pill or a surgery I can do to change that. So you need to stay away from it. And things like support groups, like AA, help with that. Help with that. What about the physical dependence model?
Can't they put you like in a drug sleep, so that way you can sleep through the
Yeah,
rest
so
of the
treatment facilities, go into a treatment facility where they can medically help you through that process. Medically help you through that process of withdrawal to get you safely to the other side, right? And then what about the positive reinforcement model?
Therapy. Avoiding.
Therapy, so that we can, I'm sorry, what?
Avoiding places so that.
Sure, so some of the cues and conditioned learning, right? But more so, what was the positive reason you were using that drug? Why were you abusing that drug? Can we resolve that situation for you so that that's no longer contributing to your desire to use the drug? All right, that's all I got. Do y'all have other questions for me about things? Yes, Catherine.
Did you say that, you were gonna add multiple choice for the last two slides? It's
I'm
just-
not. No multiple choice. All three responses. Yes, Lee.
Will you post this video?
Yes, I will. And I think almost all the lectures are posted on playlists. If you have a question about it, like, I don't remember, looking through my notes, I don't know what this means, go find the YouTube one. Not all of them are there, but I got most of them. I'm gonna take a picture of that and insert it in here. I'll take a picture of this and insert it in here. That's on the last slide. See, right now, they're next to the last slide. So
So there'll be 8 for your spots.
10, 8, 10,
And
not more than 10.
however many you give, the same for all of them, so if
No, I think there's some options.
Ah,